under a low-powered microscope lens in a laboratory where the only sound is the faint hum of equipment and the occasional shift of a chair on a tile floor. Something no larger than a grain of sand sits motionless on a fragment of dried moss. Its body is barrel-shaped, rounded at both ends, and faintly amber in color where the light catches it. 8 legs, each tipped with small claws, are folded inward and retracted so completely that they are nearly invisible. The creature looks at first like a mote of dust that happened to land in a particular way. It is not dust. It is not exactly asleep. It is something for which ordinary language does not have a good word. This is a tardigrade in its tune state. The name comes from the barrel shape, tun being an old word for a large cask, and the shape itself is not accidental. By drawing its legs inward and contracting its body into this specific form, the tardigrade has reduced the surface area it exposes to the world by a significant fraction. What is happening inside it at the moment of this observation is as close to nothing as biology can arrange. Its metabolic rate has dropped to something below 1/100th of 1% of what it normally runs. It is not breathing in any meaningful sense. Its cells have expelled most of their water and replaced it with a glassy sugar compound that holds the cell structures in suspension, not frozen, not dissolved, simply held, like insects preserved in amber, but still theoretically capable of resuming where they left off. The fragment of moss the tardigrade rests on was collected from a hillside in Antarctica. The temperature outside the building where this microscope sits is several degrees below zero, and in the place where the moss was found, the temperature can fall far lower than that. The tardigrade does not appear to register this. It does not, in any meaningful sense, register anything at all. Its nervous system is not processing signals. Its heart, which in its active state beats in a rhythm visible under magnification, is still. Whatever constitutes the experience of being a tardigrade, if such a thing can be said to exist, is at this moment entirely absent. The body remains. The life is somewhere else, or is waiting, or is held in a form that does not map cleanly onto the ordinary meanings of the word. What the tun state allows is almost implausibly broad. Temperatures near absolute zero, colder than the surface of Pluto, colder than anything that occurs naturally on Earth, do not destroy it. Temperatures exceeding 150 degrees Celsius, well above the point at which most proteins denature irreversibly, do not destroy it either. Pressures greater than those found at the deepest point of the ocean floor, where the weight of miles of water compresses everything, leave the barrel-shaped body intact. At some point in the early years of this century, a set of tardigrades was carried aboard a spacecraft and exposed directly to the vacuum of outer space, unshielded, unhoused, simply present in the hard radiation environment beyond the atmosphere for 10 days, and a significant portion of them survived to be brought back to Earth and revived. They were placed in water, they extended their legs, they moved. The recovery from the tun state is not instantaneous, but it does not take long. Given moisture and a temperature within their normal operating range, most tardigrades resume metabolic activity within a few hours. The legs extend, the body softens slightly from its barrel shape. The creature begins to move with the characteristic shuffling gait that gives the group its common name, water bear, a slow, 8-legged walk that looks, at the scale of a microscope, somewhat like a very small and deliberate animal making its way across an uneven landscape, which is exactly what it is. The fact that this same animal could be revived after a decade in a dry state, or after exposure to conditions that would obliterate most other forms of life, does not appear from the tardigrade side of the equation to require any particular acknowledgement. It simply resumes. Tardigrades were first described in 1773 by a German pastor named Johann August Ephraim Gossi, who called them kleine Wasserbären, little water bears. They have been found in every environment that has been systematically searched for them, on every continent, at every altitude, in fresh water and in marine sediment, in hot springs, in Antarctic ice cores, on the rooftops of buildings in city centers, in the gutters of medieval manuscripts kept in climate-controlled archives. Wherever there is a small amount of moisture and a thin film of organic material, there is a reasonable chance of finding them. Their bodies are so small that they exist largely beneath the threshold of unaided human perception, which may partly explain why the full strangeness of what they can do took as long as it did to come into focus. The moss fragment on the microscope slide came from a place where the wind does not stop and the snow is permanent, and the summer, when it comes, lasts only a few weeks. The tardigrade on it has been in its tune state for an unknown length of time. When it was collected and brought to the laboratory, it gave no external sign of when its dormancy began. It could have been weeks. It could have been considerably longer. Tardigrades have been revived from dried museum specimens more than 100 years old. The record is not closed on how long the tune state can persist. There is a tree frog in North America, small enough to sit on a thumb, that spends its winters in a state almost as extreme. The wood frog, found across a range that extends north of the Arctic Circle into parts of Alaska and Canada, does not migrate and does not burrow deeply. When winter arrives at the forest floor, it finds the frog beneath a layer of leaf litter, resting in the cold, and as the temperature drops below freezing, ice begins to form, not in some abstract metabolic sense, but physically, literally, in the spaces between the frog's cells. The body surface goes rigid first, then the rigidity spreads inward. The breathing stops, the heart stops. The brain, by any measure available to researchers who have attached instruments to hibernating wood frogs and watched what happens, ceases its electrical activity. The frog is, by the criteria applied to most animals, dead. Its heart does not beat. It does not respond to handling. A body that rigid at that temperature, with no cardiac or respiratory activity, would in most other animals represent an irreversible endpoint. What keeps the wood frog from that endpoint is something its liver begins producing almost immediately as the temperature drops: glucose, in quantities that would be pathological under normal circumstances, pumped into the bloodstream and carried into the cells where it acts as a biological antifreeze, disrupting the formation of intracellular ice crystals that would otherwise rupture the cell membranes beyond repair. The ice that forms fills the spaces between cells. The cells themselves are preserved in their glucose-thickened interior, held through the months of cold in a state that is not death, but is also not anything that requires a heartbeat to sustain. When spring arrives and the forest floor warms, the frog thaws from the inside out. The heart restarts, first in tentative, irregular contractions, then settling into its normal rhythm. Breathing resumes. The glucose concentration in the blood, no longer needed, disperses. Within hours, the animal is moving. Within a day or two, in years when the timing works out right, wood frogs are gathering at the edges of thawed ponds and calling a sound that has been described as somewhere between a quacking duck and a distant chorus of small voices to announce the arrival of the breeding season. The same animal that lay motionless under the leaves, heart-stopped and frozen solid, is now at the pond edge, alive in every ordinary sense of the word. The forest around a wood frog in early winter is going quiet in its own way. The trees have shed their leaves, the insects are mostly gone. The mammals that do not hibernate are ranging wider for food, and the ones that do hibernate have already found their places. The leaf litter that covers the frog is thick with the remnants of the growing season, and it holds a little more warmth than the open air above it, though not by much. There is something in the image worth sitting with, a small animal lying still beneath the first hard frost, ice forming slowly in the spaces between its cells, while the world above it goes gray and cold and the light shortens. Not struggling, not in distress, simply waiting, in the only way its body knows, for the season to pass. The wood frog's capacity for this kind of freeze tolerance has been studied with attention to what it might mean for the preservation of human tissues, including organs. The basic challenge in preserving biological material at low temperatures is the same one the frog solves: ice crystal formation. The frog's solution, flooding the cells with glucose before the freeze, is elegant in its simplicity, though it took hundreds of millions of years of selective pressure in northern forests to arrive at it. Deeper underground, in a system of limestone caves in the mountains that run along the eastern edge of the Adriatic coast, something else has been waiting. The Dinaric Alps are riddled with cave systems, many of them connected to river systems that run underground for miles before emerging at springs lower in the mountains. The water in these caves is cold and very clear and changes very little across seasons, across years, across centuries. It is, as environments go, one of the most stable that exists on land or below it, and in that stability, an animal called the ulmen has been living for an amount of time that is difficult to fix precisely, but is certainly very long. The ulm is a salamander, though it looks, at first encounter, unlike any common conception of what a salamander looks like. It is pale white or pinkish white, nearly translucent, with a faintly rosy tinge at the gill tufts, which are external, feathery, and branch outward from the sides of its head like elaborate ornaments. Its eyes are vestigial, present, visible as small dark spots beneath the skin, but largely non-functional in the perpetual darkness where it lives. Its body is slender and elongated, the limbs small and delicate in proportion to the body length, and it moves through the water with a slow, unhurried ease that matches the pace of everything else in its environment. There is no rush in a cave that has not changed temperature in living memory. What the ulm can endure in terms of food deprivation is one of the things that drew scientific attention to it. In captivity and in field observations, ulms have been documented going without food for periods measured not in weeks or months, but in years. One study tracked individuals in their cave habitat over an extended period and found that the metabolic rate of an ulm at rest is low enough that a single meal, something small, an aquatic invertebrate, a worm, can sustain it for an extended period without any meaningful loss of body condition. The mechanism is not simply the burning of fat reserves, as in many hibernating mammals. The ulm's metabolism, when food is unavailable, turns to the animal's own tissues in a measured, controlled way, a kind of internal recycling that keeps the essential systems operational while drawing on whatever the body can spare. The result is an animal that can persist for up to 10 years without feeding. 10 years. During that time, the cave around it continues as it always has. The water flows very slowly. The temperature holds at around 10 degrees Celsius. The darkness remains total. The ulm drifts occasionally, rests and waits with the particular patience of an animal whose entire evolutionary history has taken place in an environment where waiting is the most reliably successful strategy. Its longevity is in keeping with this pace. The longest-lived individual olm documented in research literature was estimated to be approaching 100 years of age. This makes it the longest-lived amphibian known by a considerable margin. A century in a cave, the same limestone walls, the same cold, clear water, the same darkness. There is something in the scale of that which resists casual summary, a single animal present in its cave pool, while the world above it passed through a full human lifetime, while generations of the human families living in the valleys outside were born and grew old, while the forests on the slopes above the cave cycled through decades of seasons, the olm below drifting. Biologists who study cave-adapted animals use a term for the suite of traits that appear repeatedly in lineages that move into cave environments and stay: regressive evolution of the eyes, reduction of pigmentation, elongation of sensory organs, slowing of metabolic rates, extension of lifespans. The olm displays all of them. What this convergence suggests is that the cave environment applies a consistent selective pressure across unrelated lineages, and that the solutions it produces are, in broad strokes, similar. The olm arrived at its particular version of those solutions over a very long time. The estimate most often cited in the literature places the lineage's separation from its surface-dwelling relatives at somewhere in the range of 15 to 20 million years ago. The caves of the Dinaric Alps are not new. The olm's tenancy in them is not a recent arrangement. The feathery external gill tufts of the olm are not merely ornamental. They are the animal's primary respiratory organ in its aquatic environment, extracting oxygen directly from the water that moves across them. Under low magnification, the individual gill filaments are visible, fine, branching structures with a surface area far larger than their appearance at a distance suggests, each one carrying a dense network of capillaries close to the surface. The color of the gills, that faint reddish quality, is the color of well-oxygenated blood, visible through tissue, with almost no pigment to obscure it. The Omzibris in this sense transparent in a way that most animals are not. Its internal workings are faintly visible from the outside, a kind of biological candor that the permanent darkness of its cave makes largely irrelevant. In the Mediterranean, something smaller is doing something stranger. The jellyfish called asterisk Turritopsis dohrnii. Asterisk is approximately 4.5 millimeters across, at its widest point, when fully mature, smaller than a small fingernail, smaller than most people's mental image of a jellyfish by several orders of magnitude. It is transparent, with a red or orange internal structure visible at its center, and a fringe of tentacles around its bell that can number up to 90 in the adult form. It floats in the open water column, and if it were not for the specific cellular behavior it exhibits under certain conditions, it would be unremarkable, one of many small medusae drifting in the warm, shallow seas of the Mediterranean and similar waters. What it does under the pressure of injury, starvation, advanced age, or environmental stress is this: it begins to sink. The bell contracts inward, the tentacles retract. The adult form, which in jellyfish is the sexually mature, free-swimming medusa stage, begins to differentiate, the specialized cells that constitute it reverting towards something like the undifferentiated state of an earlier developmental form. The animal shrinks. Over days, it reabsorbs itself into a simpler structure and settles, eventually, as something resembling a juvenile polyp, the sessile, bottom-attached form, that is, in the normal jellyfish life cycle, a much earlier stage, and then it grows again. From the polyp state, it develops as it did before, through the normal progression of jellyfish development, back to the adult medusa form, The same individual animal, not a descendant, not a regenerated copy, but the same animal which has reversed the developmental process that normally runs in only one direction and returned to an earlier state. The cellular process that makes this possible is called transdifferentiation, a process in which fully specialized cells, already committed to a particular identity, change that identity. A muscle cell becoming a nerve cell, a cell from the mature medusa becoming a cell appropriate to the polyp stage. This is not how development is supposed to work. The standard model of development describes the process as essentially irreversible. After a certain point, once a cell has committed to being a particular type of cell, it retains that identity. Asterisk Teratopsis dornii. Asterisk appears to have an arrangement in which this commitment is, under sufficiently stressful conditions, negotiable. Whether the animal is in any meaningful sense the same animal after the reversal is a question without a clean answer. Its memories, to the extent that a jellyfish has anything that can be called memories, do not appear to persist in any form that can be measured. What persists is the genetic material housed now in a body that has unmade and remade itself. The process can, in theory, repeat. An adult medusa under stress reverting to polyp, then developing again to adult, then reverting again, the cycle without a defined endpoint. Whether this occurs in the open ocean at any scale is difficult to establish, because tracking individual jellyfish of 4 millimeters in the open Mediterranean is not straightforward research. What laboratory conditions have established is that the reversal is real, that it is repeatable, and that the mechanism is a genuine example of transdifferentiation in a living animal. The jellyfish drifts, the bell contracts, somewhere in the warm blue water, at a depth where the light is still present but diffuse. A 4 millimeter animal is folding inward, becoming something earlier than what it was, and beginning again. The Mediterranean does not pause to take note of this. It simply continues. On the dried bed of a river that may have no water in it right now, in a climate zone of Central Africa where the rains come seasonally and the dry season can stretch to months, the mud has cracked in a pattern of irregular polygons. The surface is pale and hard and gives no indication that anything is happening below it. It looks like nothing. It looks like the end of a thing. Under the surface, several centimeters down in the cooled mud, something has been sealed inside a hardened cocoon and has been breathing, slowly and quietly, for what may be years. The African lungfish of the genus Protoporus ***. There are 4 species, distributed across different parts of sub-Saharan Africa, have a solution to the drying of their freshwater habitat that is as complete as a solution can be. When the water level drops and the mud begins to firm, the lungfish begins to burrow downward into the substrate. As the mud compresses around it, the fish secretes a mucus from glands in its skin, which hardens into a lining around the burrow. It continues to secrete, and the lining thickens until the fish is enclosed in a sealed chamber, a cocoon of its own making, with a narrow channel at one end that allows air to pass to the fish's primitive but functional lungs. The entrance of the channel is covered with a porous membrane that allows gas exchange while restricting moisture loss. Inside the cocoon, the lungfish folds its body and reduces its metabolic rate substantially. It draws on fat reserves. It is not, as the ulm is sometimes described, in true torpor. Its metabolic processes have not stopped, only slowed. It breathes, slowly, shallowly, but it breathes. The thin air passage connecting it to the surface remains open, and in this state, the fish can persist through the dry season or through multiple dry seasons or through an extended drought, that prevents the rains from returning on schedule. The longest experimentally confirmed period of aestivation in a laboratory lungfish is approximately 4 years. The lungfish does not enter this state with any sign of distress. The process is gradual, a slow adjustment to changing conditions rather than an emergency response. There is no threshold moment at which the fish decides it is time. The drying of the mud and the secretion of the cocoon and the reduction of metabolism occur as a smooth progression, each step enabling the next. When the rains eventually return and the water rises and softens the mud and dissolves the outer layers of the cocoon, the process reverses with similar gradualism. The fish stirs, the cocoon breaks. The lungfish emerges, somewhat thinner, and resumes its life in the water. The fossil record of lungfish is unusually rich, partly because their bony structures preserve well, and partly because their burrows fossilize at a much higher rate than the soft tissue remains of other animals. Fossilized lungfish burrows, the hardened impressions of the cocoons they made in ancient mud, have been dated to more than 380 million years ago. The same shape, the same mucus-lined chamber, the same narrow air passage at one end. The lungfish of the Devonian period, waiting out the dry season in the same posture, in what were essentially the same conditions as the lungfish in Central Africa today. The strategy has persisted through every mass extinction event in the subsequent geological record, through the evolution and extinction of the dinosaurs, through the emergence of every lineage of land, vertebrate, the entire history of life on land, having unfolded above and around an animal that simply waited, underground, in a hardened cocoon, for the water to come back. The current lungfish, the one sealed now in the cracked lake bed with the sun moving across the pale surface overhead, does not know any of this. It breathes, it waits. The mud above it is warm in the afternoon and cooler in the night, and the fish registers neither as anything requiring a response. Whatever the fish experiences, and research on the inner states of lungfish is not a deep literature. The outward behavior is one of complete stillness, a patience so total, it reads, from the outside, as the absence of experience rather than the presence of it. But the fish is alive. Its lungs move. The same arrangement that preserved its ancestors through extinctions and ice ages and continental drift is preserving it now through a dry season that to a creature with a human lifespan would feel like a very long time. In the air passage barely visible, there is a faint movement as the fish exhales, then the stillness again. The sun continues overhead. The crack in the mud surface, where the passage connects to the surface, remains open, a thin dark line in a pale and otherwise undifferentiated expanse. From above, it looks like nothing. From below, through that narrow channel, the air that reaches the fish carries with it the warmth and the faint chemical information of the world outside, a world the fish will not see again until the season changes. The lungfish lineage is old enough that its origins predate the first terrestrial vertebrates. The first animals to leave the water and become capable of life on land were in some meaningful sense doing something the lungfish had already partially anticipated, breathing air, moving through mud, surviving the gap between water and water. The lungfish never completed that transition. It stayed, remaining aquatic, remaining sealed in its cocoons when the water left it, returning to the water when it returned. It did not leave. It did not need to. The strategy that kept it alive through the Devonian dry seasons is keeping it alive now. The cocoon hardens. The air moves in and out. Slowly, the season passes overhead, as it always has, and the fish waits below with the particular patience of something that has been waiting in one form or another since before the first creature learned to walk on land. Somewhere in Central Africa right now, in a lake bed that dried some months ago, there are lungfish sealed in their cocoons below the cracked surface. They are breathing, they are waiting. They have no awareness of what they are waiting for, and no knowledge that the rains will return. But the rains will return, and the cocoons will soften, and the fish will emerge as they always have. They will have been still for so long and so completely that their return to the open water will be just that, a return, unhurried and unannounced, to a life that paused exactly where they left it. That stillness, sealed in the dark below a dry and sunlit lake bed, is older than anything that walks or flies or swims in the world above it. It has been waiting through extinctions and ice ages and the long, slow drift of continents. It will go on waiting, in the same posture, with the same patience, for as long as the rains continue to be occasional and the mud continues to dry and harden in the sun. The air passage is still open, the fish is still breathing, the surface is warm and still and quiet. In the shallow water off a stretch of seagrass, a male seahorse hangs almost motionless in the current, his body oriented upright, his small pectoral fins beating in a rapid transparent blur. Around him, drifting outward in slow spirals, dozens of creatures no larger than an eyelash are moving away into open water. Each one is complete. Each one has a curled tail and a miniature coronet and a set of fins already working against the current. The seahorse himself is still. His brood pouch, which had been rounded and distended for weeks, now lies flat against his abdomen. He holds his position in the seagrass for a long moment after the last of them has gone, as though waiting for something that does not come. Then the current shifts, the seagrass bends, and he resumes his slow vertical drift as though nothing has changed at all. The male seahorse of the genus Hippocampus does not simply guard eggs, he carries them. After a courtship that can last several days, during which the male and female entwine tails and mirror each other's movements in the water column, the female deposits her eggs directly into a pouch on the male's abdomen. Once inside, he fertilizes them himself. The pouch is not a passive container. It delivers oxygen to the developing embryos, adjusts its salt concentration to match the surrounding water as it changes with the tide, and provides compounds that suppress immune responses so the embryos are not rejected. For several weeks, the eggs develop inside him. When they are ready, the male undergoes visible muscular contractions, bending and straightening his body repeatedly in the water until the young are expelled in bursts. Some species release fewer than 10 at a time. Others release closer to 2,000, 500 in a single birthing event. The tiny figures drift upward and outward into open water, and the seahorse returns to the seagrass, and the whole process may begin again within hours. What the female does during the pregnancy is, in one narrow sense, very little. She visits the male each morning during the gestation period, the two of them spending a few minutes together in a kind of daily greeting before she departs. The investment has been transferred. The physical burden of carrying the young, maintaining their environment, and bringing them into the water is entirely his. This is not the result of any behavioral quirk or ecological accident. It is the consistent arrangement across all known species of hippocampus, somewhere between 40 and 50 of them, distributed through warm and temperate coastal waters across much of the world. Every seahorse that has ever existed was in this same way carried and delivered by a father. Some distance inland, in warmer and drier country, there is an arrangement that requires no father at all. The Komodo dragon is the largest living lizard on Earth. An adult female can reach 3 meters in length and weigh more than 70 kilograms. In the wild, she lives on the islands of Komodo, Rinca, Flores, and several smaller islands in Indonesia, in a landscape of dry savanna and scrub forest, where temperatures remain high through most of the year. For much of her life, a female Komodo dragon may have little or no contact with a male, and in a zoo enclosure or on an island where males are absent, this presents no obstacle to reproduction. In multiple documented cases, female Komodo dragons housed entirely without males have produced viable eggs, which have hatched into healthy young. The mechanism behind this is called parthenogenesis, a form of reproduction in which eggs develop without fertilization by sperm. In the Komodo dragon's case, the specific type is called terminal fusion automixis. During egg formation, the female's genetic material undergoes a kind of internal recombination, and the resulting offspring carry a combination of their mother's chromosomes. All of these offspring are male. This is a consequence of how sex chromosomes work in Komodo dragons. Females carry 2 different sex chromosomes, and the parthenogenetic process produces young that carry only one of the 2 possible combinations, one of which results in a non-viable egg, and one of which results in a male. A single female, isolated on a remote island, or in an enclosure far from her native range, could theoretically produce male offspring who would be genetically capable of mating with her, and from that pairing, a sexually reproducing population could, in principle, establish itself. The math is unusual, the possibility is genuine. There are researchers who believe something like this may have occurred on Komodo and its neighboring islands long before human observers arrived to document it. The dragon herself does none of this with any apparent awareness of what is happening. She basks, she hunts, she digs a shallow nest in the ground and deposits her eggs with the same efficiency she would bring to anything else. The eggs incubate for around 8 months, and then, in the early morning before the heat builds, small dragons begin to emerge from the soil and immediately climb the nearest tree, where they will spend most of their early lives out of reach of larger dragons, including their mother. The remarkable thing is not that the young are able to exist at all. It is how completely ordinary the process appears to the animal doing it. There is a toad in South America whose reproduction operates through an arrangement quite different from either of these, and rather more intimate than most. The Suriname toad, Pipa Pipa, is a flat, nearly rectangular animal that looks less like a toad than like a section of bark that has been folded into a loose approximation of one. It lives in murky lowland freshwater, slow rivers, seasonal pools, flooded forest across much of northern South America, and it hunts by ambush, lying motionless in the silt until something edible drifts past. It makes no sound audible to humans. It breathes through its skin as much as through its lungs. In most respects, it is a quiet and unremarkable animal living at the dim edge of attention. After mating, which involves a prolonged underwater choreography in which the male and female somersault together through the water column, the female's eggs are pressed into the skin of her back. Over the following days, the skin of her back thickens and softens around each egg, and small individual pockets form, one per egg, each one sealing the embryo inside, in its own enclosed chamber. By the time the process is complete, the female's back is a mosaic of sealed pockets, each one raised slightly above the skin surface, each one containing a developing young toad. She carries them not against her body, but within it, at least within its outermost layer. The embryos develop through all the stages of tadpole life, entirely within their individual pockets. They do not need to breathe open water or feed from the environment. Everything they require is provided through the skin of the pocket itself, which maintains a close and intimate exchange with the developing animal inside it. After several months, the exact duration varies with water temperature. Each pocket splits open and a fully formed toadlet emerges, already the shape it will keep for the rest of its life. For a few moments, the back of the female is perforated, a temporary landscape of small openings, before the skin begins its slow return to a more ordinary surface. She will carry no visible evidence of what she has done for very long. The young disperse immediately and do not return. What makes this arrangement worth dwelling on is not its strangeness, but its completeness. The young Suriname toads experience nothing of open water during their development. The world outside the pocket does not exist for them until the moment the pocket opens. Their first contact with the environment they will spend their lives in is also the moment of their independence from the body that formed them. There is a kind of economy in this. No free swimming, vulnerable larval stage. No transformation between incompatible forms. The animal that emerges is already, entirely, the animal it will be. The axolotl, by contrast, has found a different resolution to the same broad question of what form to be. It lives in the remnant canal system of Xochimilco, on the outskirts of Mexico City, in water that is shallow, cool, and not especially clean. Its historic range was already limited. A single interconnected lake system and the urban expansion of one of the largest cities on Earth has reduced that range further still, to a degree that makes the axolotl among the more endangered vertebrates currently known to be alive. In its native water, it is a small, pale, aquatic salamander, and it is unremarkable in most respects, except that it has never grown up. Most salamanders begin life as aquatic larvae, breathing through external gills, and over time undergo a metamorphosis that transforms them into terrestrial or semi-terrestrial adults, losing the gills and developing lungs capable of supporting life on land. The pterosalatl, Ambystoma mexicanum, skips this step entirely. It reaches sexual maturity, it finds a mate, it reproduces. It does all of this while still in the form that other salamanders leave behind, the larval form, with its feathery, pink, external gill plumes fanned out around its head, its fully aquatic body, its skin smooth and thin in the way of something that was never meant to encounter air. This state, in which an animal reproduces in a form that other members of its broader group would consider juvenile, is called neoteny. The axolotl's genome has not lost the instructions for metamorphosis. Researchers have demonstrated that if an axolotl is given a controlled dose of thyroid hormone, the same hormone that triggers transformation in other salamanders, it will complete the process. The gills are absorbed, the dorsal fin recedes, the skin thickens. The animal becomes, to a substantial degree, recognizable as an adult salamander of a more ordinary type. It survives this process, but it does not thrive in the same way it does in its native form, and it does not live as long. Something in the axolotl's biology, some suppression of the signal that would normally initiate the transition, keeps it permanently at a stage that its genome treats as optional. The capacity is there and has never been used. The animal has, in effect, decided that this form is enough and has kept it, generation after generation, in the muddy canals of a shrinking lake on the edge of a great city. The external gills are the most immediately noticeable feature. They extend from each side of the head in a set of 3 feathery branches, deep pink or red from the blood vessels running through them, and they move gently in response to any current in the water or to the axolotl's own slow movement. They are fragile-looking things on an animal that seems fragile overall. pale and deliberate, resting on the sandy bottom of the canal, with its legs splayed slightly outward, its wide head turned slightly to one side, its dark eyes tracking slow movement in the water above. It is an animal that has committed thoroughly and apparently without cost to remaining exactly where it is. There is a crayfish that solved a related problem with even more radical economy. The marbled crayfish, known to researchers as Procambarus virginalis, does not have a species history that extends back through geological time. It has a history that extends back to a single individual, sometime in the 1990s, in the freshwater aquarium trade in Germany. No one knows exactly when or where the founding animal appeared. What is known is that at some point, a single crayfish underwent a genetic change, likely during reproduction, that resulted in a triploid animal carrying 3 copies of each chromosome instead of the usual 2, and that this animal was able to reproduce without a mate. The marbled crayfish reproduces through apomictic parthenogenesis, producing eggs that develop into viable young without fertilization. Each of those young is genetically identical to its mother. Each of them is also female, and each of them is also capable of the same form of reproduction. From a single founding individual, the species replicates itself outward, not as sexual reproduction produces variation, but as a photocopier produces copies, one identical generation following the next, indefinitely, in principle. The founding individual is gone, but every marbled crayfish alive today is in a genetic sense the same animal. Aquarium owners in Germany who encountered the marbled crayfish found it appealing and easy to keep. It was sold, given away, and eventually released intentionally and accidentally into natural waterways. It established populations in Germany, France, the Czech Republic, Hungary, Sweden, and various other places across Europe. It was introduced to Madagascar, where it found freshwater rice paddies enormously hospitable and spread across much of the island. In Japan, it established itself in outdoor ornamental ponds. All of these populations descend from the same founding individual. All of them are, genetically, one animal distributed across several continents. They have been found in rivers, ponds, drainage ditches, and groundwater systems. They adapt apparently without difficulty because adaptation in the genetic sense is not available to them. They cannot accumulate variation through sexual recombination, but the founding individual happened to carry a genome well suited to a wide range of freshwater conditions. But the founding individual happened to carry a genome well suited to a wide range of freshwater conditions. But the founding individual happened to carry a genome well suited to a wide range of freshwater conditions. And that genome has been reproduced, unchanged, in every marbled crayfish that has existed since. What the researchers who study the marbled crayfish find most interesting is not that it spreads, but that it represents something genuinely rare, a species whose origin falls within recorded human history and was observed, at least in retrospect, in something close to real time. The question of where species come from is ordinarily the province of deep time, geological records, fossil sequences, millions of years of gradual divergence. The marbled crayfish is a species that came from a single event, in a decade within living memory, in a country with well-maintained records of the aquarium trade. The origin story is incomplete. No one saved the founding individual, no one sequenced it before it was gone, but the broad outlines are documented. A new species appeared. It spread. It is by any reasonable biological definition its own thing now. There is something quietly vertiginous about this considered at leisure. A species requires, in most standard accounts, an extended evolutionary history, a population of ancestors, generations of gradual change, accumulating over time into something distinguishable from what came before. The marbled crayfish bypassed all of this. It required one individual, one genetic event, and a global aquarium trade willing to move animals across the world. The result moves through the dim water of a European drainage ditch entirely self-sufficient, carrying its eggs in a tight clutch beneath its tail, its body olive brown and patterned in the marbled markings that gave it its common name, indistinguishable in most superficial respects from the crayfish around it, and yet genetically unlike anything that has ever existed before. The eggs beneath its tail are small and dark and pressed close together, tucked against the underside of the abdomen and held there by the small curved appendages called pleopods. When the eggs hatch, each tiny crayfish that emerges will carry the same genome its mother carries and the same genome its grandmother carried, and all the way back to a single point somewhere in the aquarium trade of 1990s Germany. The young will disperse into the water column, settle to the bottom, and begin feeding. Within a few months, they will be carrying eggs of their own. The chain does not break. It does not require anything external to continue. The marbled crayfish moves through dim water, entirely self-sufficient, its eggs tucked carefully beneath it, the ongoing quiet replication of a single original carried forward through time and water and into the dark where the pond bottom levels out and the light from the surface grows thin and the crayfish continues, unhurried and complete, along the floor of a world it has made entirely its own. The shapes that life takes in order to continue are not uniform. They are not variations on a single theme. The seahorse transfers the physical cost of carrying young to a body that did not produce the eggs and holds his position in the seagrass after the last of his thousands of offspring have drifted away and within hours is ready to receive eggs again. The Komodo dragon requires nothing external at all, and her male young emerge from their eggs on a remote island and climb immediately into the nearest tree, carrying within them a genome that came from a single female body. The Suriname toad carries her young in the surface of her own skin, sealed and separate and complete, and delivers them to the world already whole. The axolotl settles in its canal, its gill plumes moving in the slight current, permanently in the form that most of its relatives consider a beginning, having decided or been decided by something within its cells that this is the form it will keep. And the marbled crayfish moves through dim water, alone and not alone, one individual and many, carrying its eggs in the ancient way. All crayfish carry eggs in a lineage that began with a single animal in a glass tank in Germany and has been repeating itself ever since. Across ponds and rivers and rice paddies, in the dark and the shallows both, self-contained and ongoing, the same animal and the next one and the next, moving forward across the substrate, unhurried, patient, and entirely sufficient to itself. On a forested hillside in New Caledonia, in the kind of quiet that settles after rain, a crow is standing on a branch with a strip of pandanus leaf in its beak. The leaf is torn from the serrated edge of a plant that grows at the margin of the forest. The crow holds it sideways, pulling the strip in short, deliberate movements against the curve of its lower beak, trimming away the soft fibers along one side. The rough edge comes away in small pieces. What is left is narrower, stiffer, with a slight upward curve at one end that was not there before. The crow turns the strip once, assesses it in some way that does not involve stillness, and then drives the curved tip into a crack in the bark below its feet. There is a grub somewhere inside that crack. The crow works the hook in slow arcs, feeling for resistance, adjusting the angle slightly when the grub retreats. Then the motion changes. The beak closes. The crow withdraws the strip, and the grub comes with it, carried on the hook it carved from a piece of plant it found growing at the edge of its territory. New Caledonian crows do this without being taught by humans. They do it with material they select and modify themselves, with different tools for different tasks, and with a level of precision in the shaping that varies depending on what they are trying to reach. A crack that bends requires a different tip than a hole that runs straight. The crows appear to account for this before they begin. What makes this worth sitting with for a moment is not just that the crows use tools, other animals do, and in various ways, but that they manufacture them. The distinction between using an object and modifying an object so that it will do what a natural object cannot is one that researchers have long considered meaningful. The crow does not pick up a twig and push it into a hole. It decides what shape it needs and then makes that shape from available material. The gap between those two things is not trivial. Research conducted over several decades on the island has added further detail to this picture. In controlled experiments, New Caledonian crows have been presented with problems that require multiple sequential steps, finding one object, using it to reach another, using the second to finally access a food reward the first tool could not touch. The crows solve these problems. More than that, they appear to solve them by representing, in some form, the chain of steps before they begin working through it, rather than discovering each step by accident as the previous one completes. There is something else the studies found, which is harder to describe simply. In some experimental designs, the crows were allowed to watch food being hidden, or were prevented from watching, and then had to decide how thoroughly to search for it. The crows that had seen the hiding searched with less apparent effort. The crows that had not seen it searched more systematically, longer, with more attention to locations they could not verify were empty. The interpretation the researchers offered was that the crows were tracking not just what was in the environment, but what they personally knew about the environment, and behaving differently depending on whether that knowledge was direct or absent. Knowing what you know and knowing what you do not know is a capacity that most discussions of cognition treat as significant. In the crow, it appears to operate quietly, in the field, in the ordinary course of finding food in bark. The forest around the crow, as it eats its grub, is not doing anything remarkable. The trees are the same trees. The light is the same light. What is happening is simply a bird, going about its morning, using a tool it made in a way that required it to think ahead. In shallow coastal water off the island of Sulawesi in Indonesia, the seafloor in certain places is mostly bare sand, pale, flat, and open, with very little for an animal to hide behind or beneath. For a small octopus hunting in such a place, exposure is the default condition. There is no reef nearby, no rock formation, nothing that the surrounding water has not already made visible from above. The veined octopus has found a solution to this, and the solution involves planning for the future, or something that functions like it. Scattered across the sandy areas where these octopuses live, among other debris, are the discarded halves of coconut shells, washed into the sea and settled on the bottom over years. The octopus has learned to recognize these shells as useful. It has also learned something else, that a single half shell is a shelter, but that two halves assembled correctly form something more complete. And when a single half shell is found, the work of assembling two of them has not yet finished, because a second half is not always nearby. The behavior observed by researchers in 2009 involved octopuses picking up single coconut shell halves, pressing them against the underside of their bodies, and then walking across open sand, not to a shelter, but toward a chosen destination. While carrying the shell, the octopus was exposed. It was moving slowly, visibly, in an awkward gait that used 6 of its 8 arms as legs, while the remaining 2 held the shell in place. It was, in terms of immediate self-concealment, worse off than if it had left the shell where it found it. What it was doing was transporting a future shelter to a location where, presumably, another half shell could be found or was already waiting. The assembly of the 2 halves into a portable dome, the octopus inside, pulling the pieces together around itself, came later at the destination. The carrying served no immediate purpose. Its value was entirely in what came after. The distinction the researchers emphasized in their published description was the element of deferred benefit. Using a tool to do something right now is one thing. Collecting a tool and transporting it, at cost to yourself, in terms of exposure and effort, because it will be useful somewhere else at some later moment, is something different. The second requires holding a future state in mind and acting toward it. The octopus is not a vertebrate. Its nervous system is organized entirely differently from the crows or the elephants or any of the other animals whose cognition generates interest. It has no brain in the sense those animals have brains. Most of its neurons are distributed through its arms rather than concentrated in its head. And yet what it does with the coconut shell fits the definition of planning, as well as many things that description is applied to more readily. The octopus, resting on the seafloor with its collected shell, halves arranged around it, is essentially unreadable. Its expression, if that word applies at all, is fixed. Its posture is whatever posture happens to keep the shell in position. What it is doing on the inside, whether anything that resembles anticipation is present, whether the future shelter exists for it as an image or an impulse or nothing describable at all, is not something the researchers claim to know. What they described was the behavior. The behavior was enough to be taken seriously. On a coral reef, in the kind of clear, warm water where visibility extends far enough that details at a distance are still sharp, a small fish is doing something that very few fish have been documented doing, and the scientific community has not entirely agreed on what to make of it. The cleaner wrasse is approximately 10 centimeters long. Its role on the reef is specific. It tends to a cleaning station, a fixed point that larger fish visit to have parasites and dead tissue removed from their scales, gills, and even from inside their mouths. The wrasse recognizes its clients, and some evidence suggests adjusts its behavior depending on who is watching, giving better service when other potential clients are nearby to observe. This is already an unusual behavioral profile for a reef fish. What happened in a series of experiments beginning around 2018 moved beyond it considerably. Researchers fitted a small mirror panel to the wall of a research tank and introduced individual cleaner wrasse to it. The initial response was what one might expect. The fish approached, behaved toward the reflection as though it were another fish, and after a period of time stopped doing so. That by itself is not particularly unusual. What followed was the researchers placed a small colored mark on the underside of each fish's throat, a location visible in a mirror, but not visible to the fish by direct inspection. When the marked fish were returned to tanks containing mirrors, a proportion of them began to perform a specific behavior, rubbing the throat region against the substrate, against tank walls, against surfaces, behaviors that had no prior context in their behavioral repertoire, and that appeared based on their orientation relative to the mirror to be attempts to remove the mark they could see reflected. A fish pausing before a mirror, orienting to a mark on its own body, and attempting to scrape that mark off. The mirror self-recognition test was developed initially for use with great apes, and for decades it was treated as an indicator of a particular kind of self-awareness, the capacity to recognize that a reflected image corresponds to one's own body rather than to another individual. The cleaner RAS result did not sit easily inside the existing framework. The fish has a nervous system organized in ways that make the standard account of self-recognition difficult to apply directly. Whether what the RAS is doing constitutes self-recognition in the sense that term carries, when applied to chimpanzees and elephants, in some studies dolphins, is genuinely contested. What some researchers argued was that the RAS may be passing the behavioral test without the underlying, in Bruckle-Bohr, cognitive architecture. The test was originally designed to detect that it might be responding to something about the reflected image, in a way that produces mirror-appropriate behavior without necessarily involving any representation of the self as a continuous entity. Others argued that this was too convenient a dismissal, that if the behavior looks like what the test is supposed to detect, the burden of explanation falls on those who want to draw the line somewhere between the RAS and the animals already accepted as self-recognizing. The debate has continued. The RAS study generated more published commentary than most fish cognition papers. The fish itself, in the meantime, continues going about its life on the reef, picking parasites from its clients, adjusting its service when an audience is present, and occasionally, in the presence of a mirror, doing something that no one can cleanly categorize. In a laboratory in England, in the still air of a research building on a quiet afternoon, a bumblebee is approaching a small wooden ball. The ball is sitting on the surface of a circular platform. At the edge of the platform, there is a target zone, a small marked circle. Moving the ball into the target zone releases a drop of sugar solution from a hidden reservoir below. The bee does not know this yet. What the bee knows, or what the bee has been allowed to watch, is another bee doing it, a trained demonstrator rolling the ball in a specific direction across the platform surface toward the zone, collecting its reward. Bees are not generally studied for their capacity to learn complex behaviors by observation. Their learning is well documented in other contexts. They learn to navigate, to identify flowers, to communicate the direction and distance of food sources through the waggle dance. But watching another individual perform a novel physical task and then reproducing that task is a different kind of learning. It requires, at minimum, tracking what the demonstrator is doing and extracting from the observation something general enough to apply in a new performance. The observer bees in this study did learn the behavior. They rolled the ball. They collected the sugar. That result alone was considered notable enough to publish. What was noted alongside it, and what generated as much interest as the initial finding, was the direction in which the observer bees tended to differ from the demonstrators they had watched. The trained demonstrators used a particular method of moving the ball, a specific approach angle, a specific rolling motion, consistent across their trials because that was how they had learned. The observer bees, once they began performing the task themselves, did not uniformly replicate the demonstrator's method. Some of them found a more efficient route, a more direct angle, a shorter path to the target zone, fewer steps between the start position and the reward. The bees had not been shown the more efficient version. They had not been trained to optimize. They had watched a solution and then on their own arrived at a better one. The researchers were careful in how they described this. The word that appeared in the discussion was optimization. The bees had not merely imitated. Imitation would produce a copy. What the observer bees produced, in some cases, was an improvement. Whether this reflects something that deserves to be called reasoning, or whether it reflects some other process that produces similar outputs for different internal reasons, was not settled by the study. What the study settled was what the bees actually did. They watched, they performed, and they performed it better. A bumblebee is a small thing. Its brain contains fewer than 1,000,000 neurons. The total computational substrate it has available to it is minuscule by any comparison, and yet it watched another bee solve a problem it had never encountered, extracted the logic of the solution rather than just the surface form of the movements, and applied that logic in a way that arrived at a better outcome than the one it had observed. The bee collected her drop of sugar solution. The platform reset. The afternoon continued. There is a tendency, when these findings accumulate, the crow with its hook, the octopus with its coconut shell, the bee rolling a ball more efficiently than its teacher, to look for a principle that ties them together. Some organizing explanation for why cognition keeps appearing in forms that seem to exceed what the relevant animal should need. The honest answer is that the principle is not clear, and that the search for one tends to produce frameworks that fit some cases cleanly and require adjustment for others. What is clearer is that each instance was found not by looking for something dramatic, but by watching carefully what an animal actually did in ordinary circumstances and noting that the ordinary thing it was doing was harder to explain than it looked. In Kenya, on a stretch of open savanna where the dry season has left the grass pale and the ground hard underfoot, a female elephant is moving away from the rest of her herd. She is pregnant. The movement is deliberate, but unhurried. The specific quality of unhurry that large animals carry when they know where they are going, and the distance is manageable. The herd continues its own business behind her. She does not appear to be fleeing anything or responding to a disturbance in the grass. She is going somewhere. The vegetation she is heading toward is not the closest available browse. Between her current position and her apparent destination, there are trees with edible leaves and shrubs with edible stems, the kinds of things that elephants graze routinely as they move through their range. She passes them. The selection is not random, and it is not driven by proximity. What researchers studying elephant behavior in Kenya documented over a period of observation long enough to establish patterns was that pregnant females near the end of their gestation period made these kinds of detours with a frequency that was not present in non-pregnant females, making the same journeys through the same landscape. The destinations were specific plant species. In documented cases, plants including certain leafy species and particular trees with edible bark. The plants involved contain compounds that, in controlled studies, have been found to have effects on uterine muscle activity. Compounds with the capacity to influence the timing and character of labor. The pregnant elephants were not sampling broadly and encountering these plants by chance. They were selecting them specifically from landscapes that also contained other plants they did not select. And the behavior appeared almost exclusively in females approaching the end of pregnancy, not in females at earlier stages of gestation, not in males, not in females who were not pregnant at all. How this knowledge exists in the elephant is not something the studies could determine, whether it is passed from older females to younger ones. Through observation across generations, a form of behavioral inheritance that does not require any mechanism more complex than watching what your mother did when she was pregnant, or whether there is some more direct relationship between the animal's physiological state and its orientation toward particular plants is an open question. Both possibilities are consistent with what was observed, and neither has been ruled out. The elephant's range overlaps with people. Families in parts of Kenya who live near the areas where the behavior was documented had some knowledge of the same plant species as having properties relevant to labor, not identical knowledge, not the same application, but a convergence. The researcher who noted this in the published material did not claim more than the observation warranted. She noted the convergence. She left the question of what to make of it appropriately open. What the pregnant elephant is doing at the level of behavior is moving through a landscape she knows and selecting from it what she needs. Whether she knows she needs it in any sense that resembles the way a person knows they need something is not available to inspection from outside. What is available is the movement itself, the deliberate pace, the bypassed browse, the specific cluster of plants at the end of the walk, and the slow, patient way she eats what she came for before turning back toward the herd. The scale at which this kind of knowledge operates, if it is knowledge, and the researchers use that word carefully, is different from the scale at which most animal behavior is studied. It is not a single trial. It is not a response to an immediate stimulus. It is an animal navigating a landscape over the course of a pregnancy, in the direction of something that will matter when the pregnancy ends. The long arc of the behavior, measured in weeks and months rather than seconds, makes it harder to study and easier to overlook. It was noticed partly because the researchers were paying attention to what pregnant elephants specifically did, rather than looking only at the broader patterns of elephant movement. The herd, when the elephant returns, is in the same general location. The shadow of the acacia trees has moved slightly. The rest of the group continues with the business of the afternoon, feeding, resting, the smaller members of the herd moving through the gaps between the larger ones. The pregnant female rejoins them without ceremony. Cognition, in the cases described here, tends to appear in contexts where it is useful rather than in contexts where it is merely possible. The crow makes tools on hillsides where grubs are hidden in bark. The octopus collects coconut shells on seafloors where concealment is otherwise unavailable. The bee in the laboratory is doing something its relatives might do in a different form in the field. The elephant is doing on open savanna something that relates directly to what her body is preparing for. The intelligence in each case is not abstract. It is embedded in a specific ecology, applied to a specific problem, shaped by the particular circumstances of a particular animal's life. What makes each case unusual from a scientific standpoint is not that animals solve problems. They demonstrably do. What makes each case unusual is the specific character of the solving, the tool that was manufactured rather than merely used, the object that was carried toward a future rather than used immediately, the self-referential behavior that a mirror test was designed to detect, the task that was improved upon rather than copied, the landscape that was navigated with what looks remarkably like foresight. In each case, the interesting thing is one step beyond the interesting thing one might have expected. The crow is worth returning to for a moment, because the tool-making is only part of what the research found. In studies where crows were presented with multi-step problems, sequences of tasks that had to be completed in order, the birds occasionally paused before beginning, in a way that looked, to the researchers watching, like consideration. Not necessarily the same thing as consideration in a human sense, not necessarily accompanied by anything like deliberation or mental rehearsal, but a pause before action of a duration inconsistent with confusion or with searching for an entry point to the problem that preceded a sequence of steps completed without false starts or backtracking. The pause is not evidence of anything conclusive. A pause could be many things, but in the context of the other behaviors, the tool modification, the systematic search behavior that varied depending on whether the crow had witnessed the hiding, the pause adds to a picture that is, at minimum, difficult to describe without borrowing language that usually applies to something else. The researchers working with New Caledonian crows have been doing so for long enough now that the picture has had time to get complicated. Early findings were occasionally replicated and occasionally challenged by later work with slightly different methods. The debate about what exactly the crow's metacognitive abilities consist of is ongoing. This is what productive scientific disagreement looks like from the outside. A set of careful people arguing about how to describe something they have all observed, because the observation does not fit cleanly into the categories they arrived with. The bumblebee result produced a similar aftermath. Several follow-up studies were conducted after the original publication, attempting to isolate which aspect of social observation was responsible for the efficiency improvement, and whether the same effect appeared in other novel task contexts. The answers from that subsequent work are mixed enough that the original finding has not been either cleanly confirmed as representing general problem-solving capacity or cleanly reduced to a mechanism that removes the need for any interesting explanation. The field is in the position of knowing that something happened and arguing carefully about what it was. This is, in a way, a more interesting position than certainty in either direction would be. The cleaner wrasse, at last count, remains the subject of genuine scientific uncertainty. The fish does what it does. The literature has grown around it, pro and contra, in the way that contested findings in behavioral science tend to accumulate response papers. The fish is not participating in the debate. It is on the reef, doing its work, occasionally checking its reflection. The cognitive capacities of animals are, as a field, in a different position now than they were several decades ago. The default assumption in much of the 20th century was that behaviors which appeared to involve planning, self-awareness, social learning, or abstract reasoning required special explanation, that absent strong evidence for the underlying capacity, simpler mechanisms should be assumed. That assumption has not been abandoned, and it is not wrong as a methodological starting point. But the evidence demanding special explanation has accumulated to the point where the simpler mechanisms are themselves doing significant explanatory work, and the work they are doing is increasingly visible. The octopus carrying its coconut shell across open sand is not doing it because it was trained. It is not doing it because a human showed it what the shells were for. It found the shells, assessed them, and started carrying them toward a future use in a landscape that offered no other reliable shelter. The mechanism that produces this behavior, whatever it actually is, at the level of neurons and their interactions, generates something that functions like planning. Whether it requires the same substrate that planning requires in creatures with centralized vertebrate brains is a question that the current state of knowledge cannot settle. What the current state of knowledge can say is that the substrate in the octopus, however different it is, produces the output. Somewhere in the uplands of New Caledonia, in the long light of late afternoon, the crow has finished its meal. The hook tool is still in the crack, or has been dropped somewhere in the bark below, or has been retained in the crow's foot for the next probe. Research suggests they sometimes do this, carrying a successful tool to the next task rather than discarding it after one use. The forest is doing what forests do in the hour before the light changes. A few insects move through the understory. The wind, where there is wind, shifts the leaves at the edges of the canopy. The crow stands where it stood, or has moved to another branch, and is continuing in whatever way it continues, alert to the things that matter in its daily round, attuned to the landscape it knows, doing what it does with the equipment it has. In Kenya, the sky above the savannah is the particular shade it turns before the evening cools, and the pregnant elephant is back with her herd, moving at the unhurried pace of large animals that have found what they were looking for and have nothing urgent left to do before the light fails. She will eat more tonight, the usual browse, the things that are always there. What she found on the detour is already doing whatever it does inside her, quietly, without announcement. The herd moves. The dry grass makes a soft sound. The long, flat horizon holds the last of the afternoon. There is something worth staying with in the image of that elephant on her walk, not for what it proves, because what it proves is still being carefully argued about in the relevant literature, but for what it suggests, that an animal's engagement with the world around it can extend across a time frame that is not measured in the moment of hunger or the moment of threat, that the landscape can be a resource not just for what it provides right now, but for what it will matter for later, that some forms of knowledge about which plants, which properties, which needs a body is moving toward can travel through a species across generations without language, without formal instruction, without anything that announces itself as learning. The elephant does not know she is remarkable. She does not know there is any comparison being made. She knows, in whatever way she knows anything, where she was going and that she has been. The walk is finished, the herd continues, and somewhere in the clear warm water off the coast of Indonesia, a veined octopus has settled its collected coconut shell halves into position on the sandy seafloor, drawn the two pieces together around its body with slow, precise arm movements, and is now inside them, hidden still, complete in its shelter in the middle of the open sand. The ocean continues past the shell in whatever direction the current runs. The octopus is quiet. It has what it came for. A platypus moves through cold river water with its eyes shut. The nostrils are sealed. The small ear slits have closed. Every external sense has been set aside for the duration of the dive, and what remains is the bill, broad and flat and sweeping slowly from side to side through the current, reading the water in a way that has no precise equivalent in any human experience. Somewhere a few centimeters below the riverbed silt, a small shrimp shifts its position. The muscle contraction is slight. The electrical signal it generates is smaller still, a flicker of bioelectricity in the cold, dark water, faint enough that almost nothing in the world could detect it. The platypus turns, slightly, and follows it. There are roughly 40,000 electroreceptors distributed across the surface of that bill, along with approximately 60,000 mechanoreceptors that register pressure and movement in the water column itself. Together they give the platypus a map of its environment that requires no light and no sound, only the slow drift of the bill through the current and the continuous arrival of signals from every small thing moving in the dark ahead of it. Neurologists who have studied the platypus brain note that the region of the somatosensory cortex devoted to processing information from the bill takes up a disproportionate share of the whole, roughly the same proportion as the human hand occupies in the human brain, the hand being the structure we use to gather the most precise tactile information about our world. The platypus has in its bill something like a hand that works in water and reads electricity. What makes this worth sitting with is what it means for the river the platypus inhabits. That same stretch of cold water to a human observer on the bank contains gravel and current and the occasional visible fish. To the platypus moving through it, eyes closed, the water is dense with electrical signatures, the heartbeats of buried invertebrates, the faint muscular tremor of a shrimp adjusting its grip, the field generated by a small crayfish moving along the bottom. A river that appears to the human eye to contain relatively little is in the sensory experience of the platypus, a highly detailed landscape of hidden movement, the same water, an entirely different world. The platypus is native to eastern Australia and Tasmania, living in rivers and streams where the water is cold and often murky enough that vision would not be especially useful even if its eyes were open. The bill it uses to navigate this environment is soft and pliable, covered in a smooth skin that is kept moist and sensitive through the animal's time in the water. Out of the water, the platypus uses its eyes and nostrils normally. The transition between its two sensory modes happens at the surface. Entering the water, one world closes down entirely and another opens. The first Europeans to encounter a preserved platypus specimen were reportedly skeptical. The combination of a duck's bill, a beaver's tail, webbed feet, and fur struck the naturalists of the late 18th century as sufficiently improbable that the initial descriptions were thought to be fabrications. The zoologist George Shaw, examining one of the earliest specimens in 1799, reportedly checked the bill for stitches, looking for evidence that someone had sewn a duck's beak onto a mammal's body as a hoax. There were no stitches. The animal was simply what it appeared to be, which in this case was something that had not been expected. The bill was only the beginning of what made the platypus unusual. Male platypuses also carry a venomous spur on each hind leg, capable of delivering a venom that causes severe and long-lasting pain in humans and is apparently used in competition between males during breeding season. The venom contains compounds found nowhere else in any animal, proteins that appear to have been assembled into this specific chemical combination independently of any known mammalian venom. The platypus, in other words, is an animal that found its own solutions to several problems that other animals solved differently and arrived at answers no one would have predicted. The bill returns to the same patch of riverbed 3 times in one dive, sweeping across it in slightly different arcs, as if confirming what it already found. The shrimp is located. The platypus adjusts its angle of descent through the water, moving toward the place the signal came from, navigating by a sense that has no name in ordinary language, something between touch and hearing and the detection of electricity, all of it arriving through a soft, flat organ sweeping back and forth in the cold and the dark. Mantis shrimp live on tropical coral reefs, and most of what they see, no human eye can follow. They are small animals. Most species fall somewhere between 4 and 30 centimeters in length, and they spend much of their time in burrows cut into coral rubble or soft sediment, emerging to hunt or to defend their territory with a strike from their front claws that is among the fastest movements produced by any animal. The strike can happen in less than 2 milliseconds and generates a pressure wave capable of cracking the shells of snails and crabs. That part of the mantis shrimp's biology is relatively well known. The visual system is something else entirely. A human eye contains 3 types of photoreceptor cells, each sensitive to a different range of wavelengths, roughly corresponding to red, green, and blue light, with the brain combining information from all 3 to produce the full range of color perception. The mantis shrimp eye contains 16 types of photoreceptor cells, 16 distinct categories of color receptor, along with additional receptors sensitive to ultraviolet light and to circularly polarized light, a property of light that has no analog in human vision, and which the mantis shrimp both detects and produces in signals from its own body. The eyes move independently of each other, each one capable of looking in a different direction, and within each eye, the receptors are arranged in a distinct band across the middle of the visual field, rather than distributed across the whole surface. For years, the assumption was that 16 types of color receptor must mean extraordinarily fine color discrimination, the mantis shrimp seeing gradations of color that no human visual system could resolve. A study published in 2014 in the journal Science found that this was not exactly the case. When researchers tested mantis shrimp on their ability to distinguish between colors of very similar wavelengths, the animals performed considerably worse than expected, notably worse than humans at that same task of fine discrimination between similar hues. What the researchers proposed instead was that the mantis shrimp visual system may not be doing the same thing as human color vision at all. Rather than comparing signals across multiple receptor types to calculate a fine gradient of color, the shrimp may be processing color through rapid temporal scanning, running through the different receptor types in quick succession to identify what category of color is present, not what precise shade it is. A system built for fast recognition rather than subtle distinction. The difference is something like the difference between a system that determines exactly what shade of red something is, and a system that determines immediately, in a single brief moment, that it is red and not blue and not green. The mantis shrimp, moving through a reef where a correct and rapid identification of what is approaching might matter considerably, may have a visual system optimized for speed of response rather than the subtlety of distinction that human color perception allows. 16 types of photoreceptor arranged not to produce finer resolution, but to produce faster answers. The richness of the equipment put to work in a way that does not simply scale up from what we already do. On the reef, the mantis shrimp sits at the entrance of its burrow and surveys the scene with eyes that swivel in independent directions, each axis of motion controlled separately from the other. The stalked eyes can move up and down, back and forth, and rotate on their own axes, giving the animal a nearly complete sphere of vision and the ability to track separate objects with each eye simultaneously. A prey animal approaches from one direction, a potential threat appears from another. The mantis shrimp registers both without moving its body, the eyes working independently through the 16 categories of the light that fills the water around it, reading a reef that contains information the surface of which human vision barely reaches. The star-nosed mole surfaces through damp soil in the dark. It is small, about 17 centimeters long, with a plump body and short, powerful forelimbs designed for digging, and a tail that thickens in winter as the animal stores fat for the cold months. What makes it easy to identify is the structure at the center of its face, 22 small fleshy rays arranged in a ring around the nostrils, pink and constantly in motion, spreading open and contracting as the animal moves. The rays look somewhat like the petals of a small flower, or like the arms of a small sea creature, and they are in near continuous motion whenever the mole is active, tapping the ground ahead in rapid, probing pulses that return a continuous stream of tactile information about the surface being explored. Distributed across those 22 rays are approximately 25,000 tiny touch organs called Eimer's organs, each one capable of registering fine details of texture and surface structure. The whole nasal star covers roughly the same total surface area as the tip of a human fingertip, but packed into that small space is a concentration of sensory receptors that gives the star-nosed mole one of the highest densities of touch receptors found in any mammal. The corresponding region of the mole's brain, the area that processes information from the star, takes up a disproportionately large share of the total somatosensory cortex, just as the platypus bill does in its own brain. The nervous system organized around the organ that does the most work. The speed at which the star-nosed mole processes tactile information is one of the more striking things that has been documented about it. Studies using high-speed photography have measured the time from first contact with a prey item to the decision of whether to eat it at as little as 8 milliseconds. The mole touches something, determines what it is, and decides all within the span of time that most human actions take to even begin. The star is touching an area of tunnel floor ahead of the mole at a rate of up to 13 taps per second while the animal is moving, and each tap is resolved into a detailed tactile image almost instantaneously, allowing the mole to search and assess at a speed that appears, when filmed, closer to a reflex than a decision. There is something else the star-nosed mole does that is worth knowing about. It can smell underwater. This is unusual for a mammal, most of which do not have functional olfaction in a submerged environment, because the mechanism of smell requires air-carrying scent molecules to contact the olfactory epithelium, which cannot happen when the nose is full of water. The star-nosed mole has found a way around this. When moving through water in the flooded portions of its tunnel system, or while swimming in the shallow streams it frequents, the mole exhales small bubbles of air onto surfaces it is investigating, and then immediately inhales them again. The air bubble contacts the substrate, picks up any scent molecules present, and returns to the nose carrying them. The mole is in effect exhaling a temporary pocket of air-based detection equipment and then retrieving it. The result is that the star-nosed mole can hunt effectively in complete darkness, both underground and underwater, using touch and smell in a combination that covers virtually every surface it can reach. Researchers have described it as one of the fastest acting sensory systems in any mammal, a nervous system built around the expectation of constant high-speed information from the world directly in front of it. The mole moves through its tunnels with a kind of contained urgency, the star tapping ahead in rapid pulses, the whole body following the information the star generates, an animal almost entirely defined by the structure at the center of its face. In the damp cold of the tunnel, somewhere below the frost line, the star is in motion. The rays fan open against the soil and then contract, fan open again, working methodically through the patch of ground ahead, and 8 milliseconds after the star makes contact with something small and still, the mole has already decided what to do. Pit vipers hunt in complete darkness by reading heat. The two loreal pit organs that give the group its name are located between the eye and the nostril on each side of the head, small depressions in the scales that open into chambers lined with a thin membrane containing thousands of heat-sensitive nerve endings. What arrives at those membranes is infrared radiation, heat energy emitted by warm objects, including the bodies of warm-blooded prey. The system is sensitive enough to detect temperature differentials as small as 0.003 of a degree Celsius, a precision that allows the snake to locate and strike accurately at a mouse or a rabbit in conditions of complete visual darkness, detecting not just the presence of the animal's warmth, but enough spatial detail to aim. The laurel pits work in pairs, one on each side of the head, and the slight difference in the signal arriving at each side allows the snake to locate the source of heat in three-dimensional space, much as two ears allow a hearing animal to locate a sound in space by comparing the slight timing difference between signals arriving at each side. A timber rattlesnake holding still in a rocky crevice at night receives a heat image of anything warm moving within its range, not a sharp visual image, but a map of relative temperatures in the air in front of it, warm shapes moving through cool background, the profile of a mouse distinct enough to guide a strike. The snake does not need to see. In the full dark of a rocky hillside in autumn, the pit organs deliver all the spatial information the strike requires. What researchers found when they examined how this thermal information is processed in the brain was that the inputs from the pit organs and the inputs from the eyes are routed to the same region of the midbrain, the optic tectum, which in most vertebrates processes visual information. In the pit viper, this region has been co-opted to handle both visual and thermal inputs, combining them into a unified sensory map of the environment ahead of the snake. In conditions where both senses are functioning, in dim light, where the eyes can still contribute, the two streams of information are integrated into a single picture. In total darkness, the thermal map alone is sufficient. The system is built to give the snake a continuous, accurate representation of where warm-bodied things are, regardless of whether there is any light to see them by. Pit vipers are distributed across most of the world's warmer regions, with species present across Asia, the Americas, and parts of Africa. The family includes rattlesnakes, copperheads, cottonmouths, and the fer-de-lance, along with a large number of species in Asia. All of them share the laural pit structure, though the pits vary in size and sensitivity across species. Some arboreal species that hunt birds have particularly sensitive pit organs. Those that hunt primarily small mammals on the ground have a sensory map tuned to the body temperature and the size of a rodent moving through the dark. The timber rattlesnake on a rocky Pennsylvania hillside in October is largely still. It does not need to search actively. It holds its position and waits, the pit organs registering the temperature of the air, the background reading a consistent low that shifts only when something warm moves into range. A mouse foraging through the leaf litter crosses into the field of detection. The pit organs register the change, a warm shape against the cooler background, closer on the left side than the right, moving at a particular speed and in a particular direction. The snake does not need to move, or shift its coil, or lift its head. The picture arrives complete. What happens next follows from what the picture shows. The laural pits evolved in the pit viper lineage, independently from a similar structure found in some pythons and boas, which have developed their own heat-sensing organs along the jawline. The convergent evolution of heat sensing in two separate snake lineages suggests that detecting warm prey in the dark offers enough of an advantage that the required neural machinery was worth assembling twice, independently, in animals that shared a common ancestor but went their separate evolutionary ways, long before either group developed the ability to detect infrared light. In the dark of a rocky crevice, the cold body of the snake waits, and the thermal map of the world directly in front of it arrives in a steady, unhurried stream. Sharks have been reading the electrical signatures of hidden animals for at least 400 million years. The structure responsible for this is the ampullae of Lorenzini, small, jelly-filled pore organs, clustered around the snout and the underside of the head, each one consisting of a canal that opens to the surface through a pore, lined with sensory cells that respond to differences in electrical potential between the pore opening and the base of the canal. When an animal nearby generates an electrical field through its muscle activity or its nervous system, the shark's ampullae detect the change. The sensitivity of the system is considerable. Studies have measured the detection threshold at approximately 5 nanovolts per centimeter, which is among the most sensitive electroreception systems documented in any animal. A nanosystem of electrical detection so precise that it can register the heartbeat of a flatfish buried under an inch of sand from a short distance away. Sharks use this system in multiple ways. The most widely discussed is the detection of prey hidden from view. a stingray buried beneath the sand of a shallow lagoon, a flatfish lying still on the sea floor, anything with a heartbeat and a nervous system producing the faint electrical fields that biological tissue unavoidably generates. The shark's head sweeps across the seabed, and where the sand appears uniform to every optical sensor, the ampullae of Lorenzini reveal the precise location of the body below. The stingray, lying still in the hope of being overlooked, is generating the one signature the shark does not need eyes to find. There is a second function that researchers have been studying for somewhat longer, which may be more fundamental. The Earth's magnetic field produces electrical fields in seawater when the water moves through it, and a shark moving through seawater also generates a weak electrical field as its body intersects with the geomagnetic field lines. The hypothesis is that sharks can detect these fields using the ampullae of Lorenzini and use them as a form of geomagnetic compass, not reading map coordinates, but maintaining orientation relative to the Earth's field in the same way that a magnetic compass needle orients to magnetic north. Long-distance migrations, return journeys to specific breeding sites, the maintenance of a consistent heading across hundreds of kilometers of open ocean, the ampullae may be part of how these navigations are accomplished, though the exact mechanisms are still being worked through. The great hammerhead shark, whose broad, flat, wing-shaped head concentrates a large number of ampullae across its wide surface, is often described in relation to electroreception because the unusual shape of its head has led researchers to investigate whether the spread of the sensory organs might provide advantages in detection. The wide cephalofoil, sweeping from side to side as the shark moves forward, covers a much wider strip of seafloor on each pass than the narrower head of other shark species would. Over a sand flat where stingrays are resting, this translates into a more efficient scan, more ground covered per unit of forward movement. The sensory array spread wide and continuously active, reading the electrical signatures of every buried animal within range. The great hammerhead is a large shark, adults typically measuring somewhere between 3 and 4 meters in length, and it moves through shallow tropical water with a characteristic slow, side-to-side sweep of the head that makes the electroreceptive function easy to picture in action. The cephalofoil moves left, then right, then left again, covering an arc of seafloor considerably wider than the shark's body, and the ampullae distributed across its flat underside. Read each portion of that arc as the head passes over it. A stingray buried in sand that would be invisible to any visual sense leaves an electrical trace that the passing sweep of the hammerhead's head resolves into a location, a size, an orientation. The sand appears empty. The hammerhead follows the reading, not the appearance. The ampullae of Lorenzini are also sensitive to changes in water temperature and to mechanical pressure, making them a multi-purpose sensory organ that may do several different things simultaneously. The jelly that fills each canal is a conductor with unusual properties, and the sensory cells at the base respond to changes in the electrical potential across the jelly with high precision. Lorenzini himself described the organs in 1678, naming them as was conventional for himself without knowing what they were for, classifying them as some kind of sensory structure whose function he could not determine. It was not until the mid-20th century that researchers determined they were electroreceptors, and the full picture of what they detect and how continues to be refined. The hammerhead moves along the sandy bottom at what is, for an animal of its size, a relatively unhurried pace. The cephalofoil sweeps left. It sweeps right. The sand below is flat and unmarked, undisturbed by any visible presence. What the ampullae of Lorenzini are reading is something different, not the surface of the sand, but the electrical geography of what lies beneath it. A stingray buried 3 centimeters below the surface, completely still, its outline invisible from any angle. Its heart is beating, its muscles are maintaining a slight electrical tone. The field it generates extends upward through the sand and into the water column above it, and as the hammerhead cephalofoil passes overhead, the ampullae register the field with the clarity of a signal arriving in clear air. The sand flat looks empty. It is not empty. The hammerhead knows the difference because the ampullae tell it continuously what the undisturbed appearance of the seafloor actually contains. What all of this amounts to, if it is held together for a moment, is the possibility that the same patch of ground, or the same stretch of river, or the same section of reef contains a different world depending on what you bring to reading it. The river the platypus moves through has in its cold water an electrical landscape of hidden muscle contractions and small heartbeats that the platypus reads. With a bill the nervous system has organized its entire somatosensory cortex around. The reef the mantis shrimp inhabits has 16 categories of light moving through its water, along with polarized light signals from the bodies of other animals, and the mantis shrimp reads them all in a system optimized not for subtlety, but for speed. The tunnel soil the star-nosed mole pushes through has a tactile texture that resolves at the tip of 22 tapping rays into the precise location and character of every small thing within reach, assessed in milliseconds too brief to be deliberate. The darkness the pit viper waits in has, moving through it, a thermal landscape of warm bodies against cool air, the snake reading it continuously through membranes thinner than paper on each side of its head. The sand flat above which the hammerhead glides has, below its surface, a field of electrical signatures mapping every animal with a heartbeat, buried there, invisible to light, readable to jelly-filled poor organs named in the 17th century by a researcher who did not yet know what they were for. 5 environments, 5 animals, 5 animals, 5 different versions of what constitutes the world. The human sensory experience of all of these places, the river, the reef, the tunnel, the dark hillside, the sand flat, would produce a version of each that is, by comparison, thin, not wrong, but incomplete in ways that are difficult to fully appreciate, precisely because the missing categories are not accessible. It is not possible to know, from inside a 3-receptor visual system, what 16 categories of color looks like, or to understand, from skin that has never detected an electric field, what the electrical landscape of a riverbed feels like. The incompleteness is not felt as incompleteness because nothing is missing from the inside. The full range of what the platypus bill detects is simply not part of the picture. What the biology offers, looked at from a sufficient distance, is something like a reminder that the physical world contains far more information than any single sensory system extracts from it, and that the information different animals extract is not always a subset of the same signal. Circularly polarized light. Geomagnetic fields read through the electrical induction of a moving body, the heartbeat of a buried animal, the thermal profile of a mouse in complete darkness. None of these are approximations of human sensory data. They are different categories entirely, different aspects of a physical world that is richer and more multiply legible than any one creature's experience of it. The platypus has returned to the surface. It rests for a moment at the bank of the river in the early morning, the bill pressed into the mud of the bank, the eyes open now, the nostrils unsealed. In a moment it will dive again, the external senses closing down, one by one, as it enters the water, the bill beginning its slow sweep through the cold current. Below the surface, the river will be full of signals. The platypus will follow them through the dark in the particular, unhurried way it has been following them for perhaps 20 million years, reading a version of the river that has always been there and that most creatures moving through it have never found. The hammerhead makes one more pass over the sand flat, the cephalofoil sweeping slowly in a long arc from left to right, the ampullae of Lorenzini reading the electrical field of everything buried beneath the surface. The water above the sand is clear and still. The light comes in at a shallow angle and falls across the flat bottom, and to any eye looking down from the surface, the sand appears empty and smooth and uninhabited, a clean and featureless expanse of pale sediment with nothing on it but the shark moving above it in long, patient arcs. Below the sand, the hidden lives continue their electrical existence, their heartbeats and their small muscular adjustments broadcasting into the water above, each signal reaching the hammerhead's sensory array as clearly as a voice in a quiet room. The sand flat contains a map of everything alive inside it, and the hammerhead reads it continuously, passing overhead in the clear tropical water, the same patch of ground meaning something entirely different to the creature passing over it than it does to anything looking down from above. Somewhere below the surface of the earth, in a tunnel no wider than a fist, a naked mole rat is moving through the dark. The walls are packed clay and old root fiber, smooth where thousands of small bodies have passed through them over many years. The air is warm, warmer than the surface above, where the African night has cooled the grass and the soil has begun to release its heat, and in the close, still atmosphere of the tunnel system, there is the faint sound of movement in both directions, workers passing and repassing in the continuous industry of a colony that does not observe night or day. There is no light here. There has never been light here. The mole rats navigate by touch and by smell, and by the accumulated knowledge of a network of passages that may extend for several kilometers in total, a subterranean architecture built and maintained and extended by animals whose entire lived world is this warm, dark, close, productive underground. A colony of naked mole rats can hold close to 300 individuals, and the social structure that organizes them is unlike anything found in other mammals. At the center of the colony is the queen. She is physically distinct from every other female in the colony, not by some small margin, but visibly, unmistakably so. Repeated pregnancies have lengthened her torso, adding space between her vertebrae in a way that does not happen to any of the other females around her, whose bodies remain reproductively dormant for as long as she is alive. They retain the capacity to become queens. The biology is latent in each of them, but while she is present and producing offspring, that capacity stays dormant, held in check by a combination of behavioral and chemical signals that the colony generates around her presence. When she is removed, that changes. A new queen will emerge from among the workers. The body will respond, the dormant will become active, and the colony will continue. The workers themselves are not all the same. Body size in a naked mole rat colony is roughly correlated with the kind of work an individual performs. Smaller individuals tend to carry out the more frequent tasks, the tunneling, the food transport, the grooming of pups. Larger workers are less active day to day, but are more likely to respond when the colony is disturbed, moving toward the source of disruption rather than away from it. This division is not rigidly fixed in the way that caste structure works in insects, where the body's form determines its function from birth and cannot change. In naked mole rats, The role an individual performs is shaped partly by size and partly by history, by which tasks a given animal has been conditioned to perform over time. The colony is organized, but it is organized in a way that has a certain flexibility built into it, a capacity for reorganization that the colony can draw on when circumstances change. Below the packed earth ceiling, in tunnels that branch and reconnect, in patterns that have grown more complex over decades, the colony's industry continues through the night. Food is moved from the foraging tunnels toward the central chambers. Pups are tended. The queen moves through the system, and the workers part around her, and the warm air carries the chemical signals that keep the whole structure coherent, not by instruction from any one individual, but by the cumulative effect of many small exchanges, each one unremarkable on its own, together producing something ordered and enduring. A leafcutter ant colony is a different kind of order entirely, where the mole rat colony is intimate and warm, organized around a single central animal. A leafcutter colony at full size contains something in the range of 8 million individuals and operates at a scale that is closer to a city than to a family. The trail from the colony to its foraging sites can stretch for hundreds of meters through the forest understory, and along that trail, at any given moment, there are ants moving in both directions, outward toward the trees and back again toward the colony, each returning worker carrying a curved fragment of leaf held upright above her body, the fragment many times larger than she is, the column from a distance looking like a slow-moving river of green flags. The trail itself is cleared and maintained, a swept path through the leaf litter, kept open by workers who spend their time removing debris rather than carrying leaves, their function entirely dedicated to the infrastructure that makes the transport possible. The leaves themselves are not food. The leaf cutters do not eat what they carry. What they carry is a substrate, raw material for a fungal garden, maintained in chambers deep within the colony, a single species of fungus that the colony has been cultivating in an unbroken line of descent for millions of years. The fungus produces small nutritious structures that the ants eat. The ants provide the fungus with chewed leaf material, a controlled growing environment, and active management of competing organisms that would otherwise colonize the garden. Neither partner can survive without the other. The relationship is not recent. Fossil and genetic evidence places its origin somewhere around 50 million years ago, which means that leafcutter ants and their fungal cultivar have been maintaining this arrangement since long before the first grasslands appeared on Earth. Among the leaf-carrying workers, on some of the larger fragments, there are smaller ants riding. These are the minims, the smallest worker caste, riding atop the cut leaves during transport, watching for a specific kind of parasitic fly that attempts to lay its eggs on the larger workers while they are occupied carrying their loads. The minim on the leaf is a dedicated guard. She is there not to help carry, but to deter. Her presence on the leaf makes the larger worker a less viable target for the fly, which is looking for an unguarded surface and a moment of inattention. The whole system, the trail, the caste structure, the fungal garden, the riding guards, runs without any individual directing it. No ant knows the shape of the whole. Each one responds to local chemical signals, and the colony from the outside appears to act with coordinated purpose. At full production, a mature leafcutter colony can strip the leaves from a mid-sized tree in 24 hours. The columns move day and night. The garden chambers below grow and are tended. The fungus produces its structures, and the ants consume them, and the whole system hums along in a steady, impersonal productivity that has been running in one form or another since before the Eocene was over. There are other kinds of collective decision. In the Okavango Delta of Botswana, in the long early light before the heat of the day becomes strong, a pack of African wild dogs is gathered close together on a stretch of open ground, a morning rally. The kind of social gathering that typically precedes the pack's departure for the hunt. The dogs are moving among each other, touching, vocalizing in the rapid high twittering sounds that are specific to this moment of collective arousal. And in the middle of the rally, some of the dogs are sneezing. The sneeze in an African wild dog pack is not incidental. A 2017 study conducted in the Okavango observed and recorded hundreds of these rally events and found a consistent pattern. The more sneezes that occurred during a rally, the more likely the pack was to move off and begin hunting. When the total number of sneezes during a rally crossed a certain threshold, departure almost always followed. When it did not reach that threshold, the pack frequently settled back into rest. The sneezes were functioning as a collective signal, a form of vote, with each sneeze contributing to an accumulating count, and the count determining whether the group moved. What made the pattern more detailed than a simple majority rule was the effect of rank. Sneezes from dominant animals, the alpha pair especially, carried more weight. When the alpha female or alpha male sneezed, the threshold needed for departure was lower. Fewer additional sneezes were required before the pack moved. When the sneeze came from a subordinate, more were needed to reach the same effect. The vote was real, but it was weighted. The voices of lower-ranking individuals were counted but discounted, and the voices of higher-ranking individuals could shift the outcome with less total input. Whether to frame this as democratic or not depends on what democracy is taken to mean. What the study found was simply that collective departure was reliably predicted by a quorum of sneezes, and that the quorum's size depended on who was doing the sneezing. The morning rally breaks up. The pack turns and begins to move across the open floodplain, the dogs falling into a loose traveling formation, the pale grass bending around them in the early light. They chose this by sneezing. The threshold was reached, and now they are going. In the wet forests of southeastern Australia, in the dense understory where the light comes down through the canopy in slow shifting columns, a superb lyrebird is standing on a low mound of cleared earth, his tail fanned open above him into a wide, intricate arc that curves forward over his back. His tail is remarkable to look at, the outer two feathers long and curved like the arms of a lyre, the inner feathers a dense mass of fine filaments that catch and diffuse the available light. But the tail is not what is most striking about him in this moment. What is most striking is the sound he is making. The superb lyrebird is one of the most accomplished vocal mimics among birds, with a syrinx capable of reproducing the acoustic structure of sounds, with a precision that recorded spectrograms have confirmed to be very close to exact. What he reproduces is not only the songs of other birds, though he does reproduce those, accurately and in quantity. He also reproduces the sounds of his environment, the specific sounds of the place he has been living in, accumulated over years of listening. In forests where logging has occurred, lyrebirds have been documented incorporating the sounds of chainsaws, of falling trees, of the particular mechanical drone of a sawmill running in the middle distance into their courtship displays. Some of these recordings are now decades old, which means there are lyrebirds producing from their own bodies the exact sound of machinery that has not operated in their territory for 30 years. The sound persists in the population, passed from males who heard it to males who have only ever heard another lyrebird reproduce it, an acoustic memory of an event that most of the birds alive today were not present for. The females evaluate his performance. The size of the repertoire matters. The accuracy of the reproduction matters. A male who can produce a wider range of sounds and produce them with greater fidelity is demonstrating something about his age, his health, his developmental history, his attentiveness to the world around him. The chainsaw sound, the camera shutter, the car engine, these are not accidents of imitation, they are information. A female who has been listening to lyrebirds long enough knows the difference between a large, accurate repertoire and a smaller, approximate one, and she uses that difference in the choices she makes. The male stands on his mound in the fern-shadowed light, his tail spread to its full width, producing in a continuous stream the sounds of a forest that contains both ancient and recent noise, the calls of birds that live in the same valleys he does, and the mechanical sounds of a human world that has been pressing against those valleys for generations. He performs for the female in the ferns, and the female watches and listens, and the forest holds still around both of them. There is a kind of economy that runs through animal social life, not in the metaphorical sense of a system that can be described using economic language, but in the concrete, observable sense of exchanges taking place between specific individuals, with histories attached to those exchanges, and with those histories influencing future behavior. The common vampire bat, which roosts in hollow trees and in cave systems across Central and South America, provides one of the most clearly documented examples of this kind of reciprocal exchange in any non-human animal. The vampire bat feeds on blood. A single bat, on a successful night, will consume enough blood to sustain herself and may return to the roost with more food than she immediately needs. A bat who fails to find food on a given night faces a genuine physiological problem. Vampire bats can die from starvation within about 2 to 3 days, which means that a single unsuccessful night is not trivial, and 2 consecutive unsuccessful nights begin to represent real danger. In a roost of several dozen individuals, on any given night, some bats will return well-fed, and some will not. What happens when the unfed bats return is what makes the system remarkable. A well-fed bat will, in many cases, regurgitate a portion of her blood meal and transfer it to a roost mate who failed to feed. This takes time and energy. It reduces the donor's own reserves. It is, by any straightforward account, a cost to the individual who performs it. The research on this behavior, conducted over many years by Gerald Wilkinson and others, found that the distribution of these food-sharing events was not random and not simply based on genetic relatedness, though relatedness plays some role. The strongest predictor of whether a bat would share food with another specific bat was whether those 2 bats had shared food with each other before. Bats who had a history of mutual exchange were more likely to share again. Bats who had been denied by a partner they had previously shared with showed measurably reduced willingness to share with that partner in subsequent interactions. The colony is not large enough, and the nights are not varied enough for every bat to have the same experience of plenty and scarcity. In a roost where food exchange is running well, the individuals who have bad nights are buffered by the exchange network they are embedded in. In a roost where the exchange network has broken down, the same bad nights become more dangerous. The colony, understood as a social structure rather than just a collection of animals sharing a space, is part of what keeps its members alive, not as an abstract collective benefit, but as a series of specific pairwise relationships, each one with its own history, each one capable of continuing or not continuing, depending on what has passed between those 2 individuals before. In the hollow tree, In the dark before dawn, the returning bats settle back into the roost. Some are full, some are not. The exchanges that follow are quiet and brief, a small transfer that takes only moments, and then the bats are still again, hanging close together in the warmth that many bodies in one place produce, even when the tree around them is cold. The naked mole rat queen is moving through her tunnels again, the workers around her flowing to the sides of the passage as she passes, the colony reorganizing around her in the small constant ways that keep its structure intact. She has been producing offspring for years, and her body has changed in response to the demands of that lengthened, heavier, physically marked by the life she has been living. In a mammal whose biology is otherwise so strange, a cold-blooded mammal, effectively, whose metabolic rate adjusts to the temperature of its tunnel rather than maintaining a fixed internal temperature. A mammal that shows almost no cancer incidence and lives to ages far exceeding what its body size would predict. The queen's physical transformation is just one more aspect of a biology that seems, the more closely it is examined, to have found solutions to problems most mammals never solved. The workers around her are not her children, though some of them are. Many of the individuals in a large colony are offspring of the current queen, born over years of continuous reproduction. Others may be older, carried over from a previous queen's reign. All of them are oriented around her and around the maintenance of the colony as a functioning system, the tunnels, the food stores, the pup chambers, the ventilation shafts that the ants at the surface would recognize, dug by animals that have never seen what a leafcutter colony builds, finding the same engineering solution from a different direction. The leafcutter column is still moving on the forest floor, far from any of this. The trail was established before dawn, and it will still be running after dark, the ants carrying their leaf fragments in the same direction, the minimums riding the larger pieces, the fungal garden in its deep chamber growing toward the new material that the workers are delivering. In the garden chambers, the workers tend the fungus with a close, constant attention that involves removing competing organisms, maintaining moisture levels, and trimming the growing edges of the garden into shapes that maximize surface area for cultivation. None of them has a model of the whole garden in any sense that the word model can be meaningfully applied to an insect. Each one is responding to local conditions, to the chemical signals of the immediate environment, and the garden that results from millions of such responses is a precisely managed, highly productive system that has been operating in essentially the same form for longer than most genera of mammals have existed. The wild dog pack is somewhere out on the floodplain now, the morning hunt underway or perhaps already completed, the dogs moving back toward their resting site in the heat of the approaching midday. The Okavango expands and contracts with the seasonal flooding, a landscape that is sometimes dry grassland and sometimes a shallow inland sea. The animals of the delta adjusting to the shape the water gives the world in any given season. The wild dogs have territories that shift with the water, and the pack moves through them with the accumulated knowledge of many hunts, many seasons, many decisions made by sneezing on the morning grass. In the forest, the lyrebird has folded his tail and moved back into the understory, where the light is lower and the leaf litter is deep and soft. He walks with the deliberate, steady gait of a bird that spends most of its time on the ground, probing for invertebrates, listening to the forest around him, and adding to the internal catalog that will come out again the next time he displays. The sounds accumulate over years. A male lyrebird can live for more than 20 years in the wild, which means the catalog built up over a long life is extensive. The sounds of many seasons, many individual birds, and machines and human presences, layered and cross-referenced in a way that the bird probably does not consciously organize, but that produces, in performance, something that sounds like order. What the female heard this morning was the compressed history of a particular piece of forest, delivered by a bird who has been listening to it his entire life. She made her assessment and is now somewhere in the ferns, going about the rest of her day. The male is also going about the rest of his. The display is over for now. The forest has gone back to its ordinary sounds. The vampire bats will not emerge until well after dark. The hollow tree is quiet during the day, the bats hanging still, their metabolic rates lowered in the resting state, the colony breathing slowly in the closed air of the tree's interior. Outside, the day passes in the way days pass in the bat's tropical range, hot, noisy with insects, the vegetation cycling through its own photosynthetic business, without reference to the social economy taking place in the dark just above the root line. At nightfall, they will emerge in sequence, spreading out across the feeding range, some finding their prey and some not, and the distribution of outcomes across the roost will once again create the conditions for the exchange network to operate. The bat who returns full will encounter the bat who returned empty, and what passes between them will depend on what has passed between them before. There is a particular quality to the social lives of animals that operate through reciprocity, through the tracking of individual exchanges over time. It requires a kind of memory that is neither abstract nor general. The vampire bat is not keeping an account of what she is owed in any explicit sense. She is responding to the history of her specific relationships, which has been encoded in some form in her nervous system and which shapes her behavior in ways that produce, across the whole colony, the stable exchange network that keeps the roost alive through the variable nights of the year. The tracking is real. The consequences of the tracking are real. Whether to call it something beyond that is a question the bats have no interest in. In the way that animal societies are built, in the underground warmth of the mole rat colony, in the long columns of leafcutter ants moving through their stripped and tended trails, in the rally and departure of the wild dog pack, in the acoustic accumulation of the lyrebird's decades of listening, in the small, careful transfers of blood in the dark of a hollow tree. There is a kind of continuity that is very old and very specific at the same time. Each of these systems has a history that stretches back beyond any individual, and a present that is maintained by the actions of individuals who will not outlast the system they are part of. The colony continues when the queen dies. The ant trail is re-cleared when the debris accumulates. The pack that disperses into new territories takes its decision-making protocols with it. The lyrebird's repertoire passes into the next generation through the patient process of young males listening to older ones. The mole rat tunnels are deep and warm and quiet. In the main chamber, near the queen, a small cluster of workers is attending to a litter of pups, blind, hairless, too new to do anything but require warmth and feeding. The workers move around them with the efficiency of animals who have performed this task many times, the colony directing its resources toward the new generation in the same steady way it directs its resources toward everything else. The pups will grow. Some of them will become workers of one subcaste or another, and some will remain in the colony's lower ranks for years, and the life of the colony will carry them along. A few, statistically, will live long enough to see the current queen replaced, and will be present for the reorganization that follows. The leafcutter minims are riding on their leaf fragments in the dark of the tunnel below the forest floor, the column descending now toward the garden chambers, the ambient smell of the living fungus intensifying as they go deeper. The garden is a pale mass of fine threads and small structures in its sealed chamber, maintained at the precise humidity the fungus requires, the temperature held steady by the insulating properties of the earth around it. The workers who tend it have no picture of its scale or its history. They have their local signals, their immediate tasks. The whole is assembled from what each of them does without any of them knowing what the whole looks like. In Botswana, somewhere in the long grass, the wild dogs are resting in the heat of the afternoon, pressed close together in the shade of a low tree, their thin legs folded, their breath slowing in the heat. Some of them are sleeping. The morning hunt is settled and distant now, and the afternoon is long, and the pack lies in its quiet collective warmth, breathing in the same rhythm that packs of wild dogs have been breathing in the shade of low trees for as long as wild dogs have been wild dogs in this kind of country. The lyrebird walks through the leaf litter in his stretch of wet forest, each footfall quiet on the deep soft ground. Somewhere in the middle distance another male is running through part of his repertoire, the calls of several other bird species, layered and alternated with a section of something mechanical, a recurring pattern that sounds, from a distance, like a camera or a mechanical shutter of some kind. The lyrebird walking through the litter pauses for a moment, apparently attending to the sound, and then moves on. And in the hollow tree, as the afternoon light moves across the bark outside, and the air within the roost stays cool and still, the bats hang in the darkness in their close, warm cluster, waiting without intention for the dark that will release them. They are small. Each one, held in the hand, would weigh less than a common apple. Their wings are folded, their small faces are pointed, in the sleeping arrangement of the roost, into the common warmth of the group. The ones who returned full last night, and shared rest alongside the ones who returned empty, and received. In the darkness of the tree, they are indistinguishable from each other, all part of the same warm breathing mass, all waiting for the same dark. The obligations between them are not visible. What passes between two bats who have a history of exchange looks from the outside like a brief contact, a transfer, a moment of stillness, and then ordinary roosting again. The record of it is nowhere you can point to, not in the tree, not in the dark, only in the bats themselves. In whatever form their nervous systems use to encode the history of specific relationships, the accumulated record of who gave and who received and when, kept quietly and used nightly, generation after generation, in a tree that has been here longer than any of the bats currently living in it, and will likely be here after all of them are gone. Outside the tree, the forest is making its slow passage through the afternoon. The light is changing. The insects are doing what the insects of this region do in the hours before dark. The air is warm and still, and full of the ordinary productive business of a tropical afternoon, and the tree is part of it, a hollow in a piece of wood, with a colony inside, living by its nightly exchange of small necessities, too small to see from any distance, too specific and particular to generalize about, except in the most careful terms. Night comes slowly in a forest. The light withdraws from the canopy first, and the understory dims before the sky above the trees is fully dark. In this interval, the colony stirs, the bats shift against each other, the ventilation of the roost changes as bodies adjust their positions, and then, one by one, and in small clusters, they emerge into the first dark air of the night, spreading out on their thin wings into the forest, going their separate ways toward whatever the night will produce. Some will return full, some will return empty. The tree will be waiting, warm with the heat of many bodies, and the exchanges that the night's outcomes require will happen quietly in the dark, as they have happened on every other night, in one form or another, for as long as this kind of animal has been living this kind of life.